This is TWIS, This Week in Science, episode number 602, recorded on Wednesday, January 18th, 2017. Live long because science! Hey everyone, I'm Dr. Kiki, and tonight we are going to fill your heads with migratory eels, menopausal whales, and early Americans. But first...
Disclaimer. Throughout the ages, mankind has sought to acquire many things: land, loot, notoriety, and the luxury of free time. But in all of our pursuits, there's one that has made us most distinctly dominant in the world: our desire and drive to acquire knowledge. And as we've acquired it, we have at times shared it, and at others kept it secret. We have hoarded it in libraries and archived it online. We created institutions dedicated to teaching what we have learned to the next generations of knowledge seekers. And we made these institutions free to democratize education. We did this as a nation founded by educated people because we knew that knowledge, above any other pursuit, would make this nation strong. And while the political talk has begun about undemocratizing our education, knowledge in the information age remains free. And as we acquire it here, it will be shared with you as always. For as long as you seek it, we will help you find This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
It's science to Kiki and Blair.
And good science to you, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're now well into the 600s. You know.
Oh yeah, just 2 episodes in. Oh, we're practically halfway to 700 at this point.
Practically there. That's right. Oh, another week into January. I was talking to someone today, and they're like, "Oh yeah, let's get something on the calendar for two Wednesdays from now. Yeah, February 1st." And I was like, "Oh my goodness!" The perception of time, it just flows onward to the sea, like all the water that has rushed through California in the last week or so. Yeah, we are back again with more science news. I have a lot. I've got a whale of a tale to give you pause. Eels elucidated and calorie restriction. What you got, Justin?
I've got even earliest Americans, from like way earlier than our earliest Americans, in fact. I've got prehistoric pet rocks and a persistent infection.
Oh,
it ain't
boy.
all bad.
Little TMI. Blair, how is the animal corner shaping up for us?
If you thought that was TMI, I have parasites, I have worms, and I have some gross spider silk.
Okay, that was actually the least gross of the bunch.
Yeah, the first couple, yeah. Stacking up there. Oh my goodness. Well, don't we have quite the show planned for you all. Don't forget, if you are in the Bay Area, you can still get tickets if you are interested for the SF Sketchfest Cal Academy of Sciences Nightlife, where we will be performing live tomorrow night.
When's that?
Tomorrow night.
Oh,
I could have used a little more notice on that. But, all right.
Yeah, so we're doing our show podcast tonight, but tomorrow night, the 19th, we are going to be live at the Cal Academy doing what we do best, except maybe it's a little bit, more over the edge for our adult audience. I don't know. We'll see. We will see. But if you're interested, you can go to the Cal Academy Nightlife website, and check it out if you are interested. We hope to see you at the Academy. And now on to the news. On to the news. So we talk about maybe recently, especially cultural differences. Right? What makes people different? How much of the DNA is responsible for that? And how much is actually other stuff going on to actually really make people, say, respond to drugs differently? Why do certain populations of people respond to certain drugs one way while other populations of people respond to the same drug in a completely different way? These are very important questions for human health. And so what, Justin, what?
I want to guess.
Well, okay, but I was going to get to it, but go ahead. Go ahead.
Well, but it's more fun if I guess before. If I guess after, it's kind of, not as
post hoc.
Guess.
Exactly. Yeah.
I will say it's going to be the microbiome, but there could be other stuff. But that would be my guess.
Well, maybe the microbiome is involved, but this study did not find that. This is a study out of UC San Francisco. I really like this study because we're going to be in San Francisco tomorrow night. Yay, or I am. Blair's already there. But anyway, UC San Francisco, they studied DNA methylation. And so we understand methylation is kind of this annotation, this slight external alteration to the DNA that takes place that determines how genes get transcribed and turned into proteins, whether or not they get turned into proteins, how much of them if they get turned on to make a lot of protein or a little bit of protein, or what exactly they're supposed to do. So
sort of the Lamarckian effect.
It is kind of, yes, this is epigenetics. This is the stuff that's on top of, outside of the genes.
Things that affect your genetic blueprint in real time as you live out a life on the planet, interacting with it. Exactly.
So things like diet or stress or stuff in your environment, stuff that's in the water, stuff that you inhale in the air, all of these things can affect you and your body through your epigenome, can change the way that you respond to things. And this study wanted to examine whether or not there were specific signatures reflected in the epigenome of an ethnic group's culture and environment, something that goes beyond the genetic ancestry. And so this is an open access article published in eLife, and what they were able to do is take a look at data from a group of Latino children, and they were able to determine whether or not there was a difference in methylation associated with either Mexican or Puerto Rican ethnicity. And they determined that 3/4 of the epigenetic difference between the two subgroups could be accounted for by genetic ancestry. So this is stuff that these are epigenetic signatures that are actually passed down from generation to generation. This is like 3/4 of it is basically, this is epigenetic genetics.
This is what you got. You're stuck with it.
But up to a quarter of the differences might actually reflect experience, practice, environment. And so this is one of the first times that we've really seen that there is this this social construct of race and ethnicity and how you live your life, how you're raised, the habits you have that may affect you biologically.
This sounds quite Lamarckian.
It does, right? Well, I mean, the issue, what's at issue here is whether or not these Lamarckian, but whether or not these epigenetic signatures, whether this methylation is something that goes on to affect subsequent generations. And that's not necessarily, that's not known. That's not understood yet. So we don't know how that actually gets transcribed and passed down exactly.
Well, but we have seen cases of it in the past. I mean, this is sort of how epigenetics sort of started to become a field. is that they would look at generations of, oh gosh, it was like Norwegian or Swedish farmers or something, and they could track like the feast and famine times of prepubescent grandparents to health outcomes in the grandchildren. And there were direct connections, whether it was a feast time or a famine time. And so these epigenetic changes have, at least in the past, maybe not in this study, but in the past, shown multi-generational changes, but not necessarily. Yeah. But then again, maybe that gets passed on to the grandkids, but then you go back to, the same baseline again. If you're talking about those grandkids, it may not be a continual effect. It may continue to reset. So both,
right. Yeah. It could. And so, there are questions. So environmental factors. If a mother smokes while she's pregnant, we know that there are health effects there on the offspring, but are there epigenetic changes that then influence the risk for further health problems? And this is kind of what they're trying to tease apart here. And so they looked at 573 children of these self-identified ethnic groups and discovered that it was basically this 25%, at least a quarter, is this
your environment,
your environment, or these kids' environment. And so the researchers say, like a standard family history, ethnicity is associated with disease for both genetic and environmental reasons. If your dad or mom had a heart attack, that tells doctors a lot about your risk for a heart attack. Part of that is genetic, but part of it is that your lifestyle is influenced heavily by your parents' lifestyle. Your ethnic group is like a much bigger family. It's partly a matter of genetics, but it also reflects the environment of your broader community. So, very, very
Once again, we hear that it's nature and nurture, folks. Nature and
right.
nurture.
Right.
It's actually three. It's nature, nurture, and Nietzsche, which is often left out of the.
Awesome. All right, moving onward. Now that we know, our environment can affect us, right? How about we go on a diet, huh?
It's January. It's time for diets.
Yeah. Well, if you restrict your calories, at least maybe as an adult, not as a child, but as an older adult, it might help you live longer.
Yes.
Yeah. So we had reported a couple of years ago on these two studies that took place, one from the University of Wisconsin-Madison and one from the National Institute on Aging, both of them looking at primates to determine how caloric restriction affects longevity. It's just a lot of rodent studies, lots of fruit fly studies, not so many primate studies. And so these two studies were supposed to be the big telling studies to let us know, all right, if we calorie restrict ourselves, will we be able to live longer? And then they came out and kind of had opposite results. Like one group, their monkeys lived a really long time. The other, they didn't really live a long time, but they had good health. And so it was like, oh, maybe it won't let you live longer, but you'll have good health. And the other one was like, nah. And so they had these conflicting outcomes. And so it's time to go back to the drawing board. And these two research teams have now published in Nature Communications the result of their revisiting of the data, going back and taking a look at the data from almost 200 monkeys from both studies to see what actually happened and to put it all together and tease out all of the things that affected it. So first, they found out the animals in the two studies had their diets restricted at different ages. So one of them, they started younger, and another, they started when they were a little bit older. And so their analysis finds that eating less is good when you're an adult. So older primates, humans even, as an adult, if you restrict your diet, it could be beneficial, but not for younger animals. Probably because it does make
developing.
Sense.
They need all the fuel.
Yeah, they're using so many calories building a human.
And then they also, and so this was different from other rodent studies, because rodent studies had found that ending earlier is better or ending or starting caloric restriction earlier is better. And then in the second point is that in the old onset group, and this was the group of monkeys at the National Institute of Aging, the control monkeys actually ate less than the control monkeys at the Wisconsin group. And this lower food intake improved survival compared to the Wisconsin control.
So this is when they could eat whatever they wanted. The Wisconsin monkeys ate more.
Yes, and part of this was it's maybe
cold.
colder, maybe.
That's what I was wondering. Colder, yeah,
But
absolutely.
part of it was also what the diet was made up of, and so the
cheese, lots of cheese in
National
Wisconsin.
Institute of Aging monkeys ate naturally sourced foods, so they had like real food, and the Wisconsin monkeys had processed food with a very high sugar content.
There we go.
And so the Wisconsin monkeys were actually fatter than the Institute of Aging monkeys.
Study on diet.
Yeah.
But,
and the one thing we're not going to control for is
sugar,
what the
fat.
subjects are eating.
Yeah, just calories. Doesn't matter.
Yeah.
So so they identified there were also sex differences in the relationship. So females were able to bear the effects, the bad effects of diet and increased fat and insulin sensitivity. So putting on weight was worse for males than it was for females. And this could be translatable to humans as well, with incidence of diabetes being higher in men than in women, and cardiovascular disease, etc. And that could have something to do with just women's physiology for carrying children. Right. That might be something that our bodies are just made up to be a little bit heavier.
Yeah. Oh, absolutely.
Okay, yeah.
For sure.
Yeah. So anyway, these are not humans, remember, but they're primates. You know, nice monkeys. Good little monkeys.
I mean, it sounds like you're saying that dieting is good for you.
It's
no,
I'm not saying
different.
that, but yeah, it's different. But it's this caloric restriction, and it's something we've been covering for a pretty long time, where it seems to increase longevity. And in this particular study, I mean, we haven't worked really this level with humans, but with primates, it does seem to be it does seem to affect aging. So it could impact longevity and health.
Right. The nice thing about this study is that since it was done on monkeys, it's something that if you do this with humans, there's so many confounding variables. If somebody's dieting, they might also be exercising. There's so much else going on. So this really allowed you to isolate specifically calorie restriction, not the type of calories, not it coupled with exercise, not any of these other things. It's all just less calories of the same food.
Yeah.
And which, if we took this show on the road 24/7, I would likely live forever.
Oh, because you'd never eat. We would stop at like all night restaurants, and all you'd order would be a cup of coffee.
This is like the refrain from Blair every time we've taken a trip. Don't you eat? Don't you ever eat?
Yes. Hey guys, Justin doesn't eat food, turns out.
It turns out he's a vampire, doesn't eat food. Nope. I mean, some people naturally don't need to eat a lot. Other people, I go through phases.
Yeah.
Right now, I'm really enjoying wintertime and eating. I'm really enjoying it.
Yeah, we need to insulate. It's cold out here on the West Coast for once.
I may be naturally doing this experiment to some extent. I don't like count my calories or anything, but I don't like do multiple meals a day. I'm good with like dinner, and that's it. Like I don't need a breakfast or a lunch. Give me a good dinner. I'm good till tomorrow night's dinner. Generally,
I can't imagine that. I eat 3 square meals a day.
Well, maybe I'll get there someday.
That doesn't sound like any fun.
No,
it's a blast. I don't know.
Yeah, I mean, if I had to calorie restrict for the rest of my foreseeable life, I would.
Yeah,
could, but it's not something that That's something I'm aiming for doing.
Cheese is just too delicious.
Because cheese and bread. Or, I mean,
And
even
chocolate.
sugar and bread.
And wine.
And chocolate and wine. Yeah.
And all of those things are so like I might have my meal might be 4,000 calories. Like I might not actually be calorie restricting at all. It's
and something that we haven't determined here is whether or not calorie restriction affects different ethnicities differently.
Diet. We'll never get to the bottom. And then does your gut microbes care? Do they need like multiple meals a day, or are my gut microbes just like, no, we're good. No, carry on. Just keep going. Like we don't even know what to do. We're still digesting. Who knows? Hey, I got a question for you.
What?
When did the first humans arrive in America? Wasn't it 9,000 years ago? 10,000 years ago? 13,000 years ago? 14,000 years ago?
Wasn't it 14,000 years ago?
None of the above.
Wasn't it 14,000 years ago?
You're both right. So 14,000 years is kind of the agreed time frame. And it's a time frame because they found the Clovis mounds. They've dated those to 13,000 years. There are a lot of outliers, though. There's a lot of sites in South America, especially, which they seem to be dating potentially to earlier than that. There's the Meadowcroft Rock Shelter site in Pennsylvania. That's been dated to between 16 and 19,000 years ago that this area was inhabited. But often these outliers become controversial and remain in a sort of debate and review stasis, because we have these solid agreements that 14,000 years is probably when everybody came over. So they keep looking for ways to tear down the older dates. Now, this is according to Université de Montreal's Department of Anthropology. And when I say they're trying to tear them down, it's because you're making a more extreme claim. And so it comes and it's an outlier of the data. There's lots of points that show 13,000 year Clovis mounds from different areas. So when you have a single outlier, it requires a lot of evidence to have it accepted, which is as it should be. And there's multiples of these, as I say. There's a lot of these in South America and that one I referred to in North America. But this is a Canadian University of Montreal's Department of Anthropology has made their recent finding is putting that date to 24,000 years ago.
What?
What? They made their discovery using artifacts from Bluefish Caves, located on the banks of the Bluefish River in northern Yukon near the Alaskan border. The actual site was excavated between 1977 and 1987. Based on radiocarbon dating of animal bones, research archaeologists back then made the then and still now bold hypothesis that human settlement in the region dated as far back as 30,000 years. At the time of the Trans-Bering Strait footbridge trek, when I was, when I first became aware of this, they were talking like 7,000 to 9,000 years ago. Right, that's when they were sort of initially putting this estimate, and that's what it would have been back in the late 70s. So saying 30,000 years, it had to be wrong. It just had to be. That's silly Canadian. In the absence of other sites of similar age, archaeologist Jacques Cinq-Mars, who did the original hypothesis, his whole thing remained controversial in the scientific community, and that's why most people haven't really heard much about it. And there wasn't a whole lot of
fringe ideas don't get so much coverage.
Yeah, and he did some radiocarbon dating stuff back then, but there were thousands and thousands of bone fragments, and he hadn't isolated and shown evidence of human activity in the mammoth, horse, bison, and caribou bones that they were finding in the caves. Right. Enter Ariane Burke, professor, University of Montreal's Department of Anthropology, her doctoral student, Lauriane Bourgeon, and a contribution also from Dr. Thomas Higham, Deputy Director of Oxford University's Radiocarbon Accelerator Unit. They looked at approximately 36,000 bone fragments that were pulled from the site and preserved at the Canadian Museum of History. Let's see who got stuck with this. It looks like the doctoral student Lorianne got stuck with 2 years of looking at bones. Her comprehensive
I'm sure
analysis,
she loved it.
Oh, absolutely. What a fantastic job, right? What they found was a small percentage of the bones that they could find traces of what can only be human activity. Series of straight V-shaped lines on the surface of the bones were made by stone tools used to skin the animals, says Burke. These are indisputable cut marks created by humans. These marks appear in distinct areas on the bones. They're characteristic of the stone tools and spots that are used to remove hard-to-remove meat and tendon. And these are unlike natural markings or animal gnawing marks. And they found them in these rooting carvings over lots and lots and lots of the specimens, right? And you know, we're talking maybe it's like 30 or 40. It's a like 1 or 2%. And one of the caves, there was 2 actual caves that they were looking at. One cave, some of the markings in the radiocarbon dating came back to about 12,000 years ago, as recent as, right? And in the other, a lot of it, there were a lot more markings and they were a lot older. So they submitted these bones for further radiocarbon dating. The oldest fragment, a horse mandible showing the marks of stone tool, apparently used to remove the tongue, was radiocarbon dated at 19,650 years, which is the equivalent of being between 23 and 24,000 years ago. There's a discrepancy here, and that's because when you do radiocarbon dating, the actual age of the bones is typically older than the radiocarbon date, because the fossil or whatever it is,
the fossilization process, as it takes in more,
you get more and more carbons getting taken up over time. So, but they have a set amount. They sort of, it's a known factor, and they know if it's this many years ago, you add this many more years, right? It's a sort of back. Anyway, they've got a formula for this. So the initial 19,650 gets pushed back to 23,000 to 24,000 years of the actual artifact being there, adjusting for its radiocarbon uptake. Quoty voice, our discovery confirms previous analysis and demonstrates that this is the earliest known site of human settlement in Canada. Well, it's not just Canada, Burke. It's all of the Americas, but yeah, Canada also. And it shows it was inhabited in the last ice age, but it was at the height of the ice age. Like, this spot is in the highest upper limit of that ice age. And they think because this is before people started traveling down into the Americas, they may have kind of been stuck there, or may have found a kind of sweet spot in this glacial age in these caves to still hunt and find game without having to venture too far from the warmth of the cave.
So I have a question.
Mm-hmm.
If this dating is done by tool marks on a skull,
mm-hmm.
is there any chance that it was not done by hominids?
That's a fantastic question. So they don't know. And this is was this one of my predictions? Did I do this prediction this year? We'd find evidence of Neanderthal in North America. I think I might have done that one. That's possible. That's totally possible. And that's something that can't quite be ruled out, except that if you take this radiocarbon dating and you look at where it is, and I mean, if you look at where this inhabitation period is of thousands of individuals, they expect it must have been there at this time. And then as deglaciation takes place, it is immediately followed by the spread of peoples to South America along the coastal routes and along routes in which the deglaciation was taking place. So it's a pretty direct, clear line from there was a people here, the glaciers went down, the ice went down, and then they traveled, and now they populate North and South America.
Okay, so there's other things pointing to it making sense for it to be that.
And if you start to take some of those outliers, like the Pennsylvania rock shelter site, that's 19,000 years, but it's so far out of the rest of the data that it's sort of looked at skeptically, and pinpoint a period directly before that. Now even those outliers seem to be falling into place for the population of the Americans.
But what if it's not even a bipedal mammal?
Well, no. So the
Like
marking,
like we were talking about we were talking about the evidence of monkeys using tools.
Using tools, but not you. I mean, this is these stone markings that are these cuts that are consistent with using stone tools on bone. Aren't being inferred for the first time in these fossils. This is a pattern. These patterns they have seen dating all the way back to many thousands of years before, as we reached the Americas. Before we reached the Americas, I mean, this is an
so
ancient.
It's the pattern and
trait.
type of the scratches that pretty clearly point to.
Yeah, you know where it's hard
things.
to remove the bit of meat or tendon, and etching away with direct marks, and these V-shaped marks are also sort of apparent that they're man-made, because they're not like just a bunch of random cuts next to each other that are forming a V. It's a straight one line here, it's a straight one line in the other direction to sort of saw into and remove bits of flesh to feast on.
Well, now that we're talking about removing bits of flesh for a feast, you know what time it is?
Oh, what time is it?
I think it's time for Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels.
What you got, Blair?
Oh, I have some parasites, you guys. Speaking of flesh. Yeah. So parasites. We talk a lot about parasites and how they're bad. You don't want a parasite, do you? Do you? No. But recent research from UC Berkeley, a full-on review looking at parasites and climate change, indicate that if we lose our parasites, we could all be in big trouble. Previous research has shown that parasites are up to 10 times more vulnerable to extinction than their hosts. And in this new study, they looked at parasites' likelihood to face extinction due to climate change compared to other taxonomic groups. And they found that losing parasites could potentially destabilize entire ecosystems by increasing other diseases, altering the food web, or changing host physiology. So they found that these parasites in hosts with variable internal temperatures, parasites of large-bodied hosts, host-specific parasites, and parasites with complex life cycles were most likely to be super vulnerable to extinction from climate change. So this is the very first comprehensive review of how climate change may affect parasite biodiversity, specifically from the point of parasite conservation, not looking at, yay, it might kill some parasites. So instead looking at actually what may be the result of this.
I'm confused. Yeah. Parasite conservation?
Yes, that's correct.
I mean, seriously?
Seriously, parasites are part of a working ecosystem.
Yeah, kind of.
Yeah, and without them, this might not be true for all parasites, but I would say most parasites serve some sort of function in their ecosystem.
Yeah, as for instance, Toxoplasma gondii functions in that it keeps would-be stray and feral cats living in a house of a woman.
I would say more that who's
willing to take on 13 to 20 needy felines.
So toxoplasmosis actually drives prey to have erratic behavior.
Right,
This
so they
could
get
be
eaten.
beneficial to an ecosystem because it reduces the prey population, and prey populations are likely to explode past carrying capacity and cause a collapse.
Absolutely.
So this could be just to look at T. gondii, it's a population control. In other situations, it could have to do with water clarity. There are lots of things that parasites can give to us that we may not realize. And that's kind of the point of this study, was looking at parasites that have very important roles in ecosystems, whether we realize it or not, and looking at their climactic tolerance. And this study predicts that parasite extinction would be clustered in particular ecosystems, and this is a call to arms for more parasite research, believe it or not, from this perspective. Not the perspective of ridding yourself of parasites, but from the perspective of seeing what function they play in an ecosystem and what their vulnerabilities are. So I think it's an interesting point of view that we don't talk about much on the show. Just like last week, I talked about how mosquitoes were pollinating.
Right, which is something great. We need more pollinators, especially if bees are dying.
And we've talked a million times on the show about how mosquitoes serve no purpose and they should just go away. But if they're pollinators, that's huge. Parasites, these are things that maybe we want them to just go away. But if they do, that opens the door to all sorts of other issues. One of the things mentioned in here was that without parasites, other diseases may surge ahead and cause more damage. So
there's also people who try to attempt to treat immune response issues, autoimmune disorders with parasites, with hookworms or some sort of because they do something in the body, and they promote induction regulatory T cells and inhibit the function of antigen presenting cells. So I mean, and I've heard it. I think we've talked about this on the show before. So there's there could be certain diseases that may present themselves because we're not carrying a load of parasites that used to counter them. I mean, we might find this is much like the microbiome. Like, there's certain diseases that seem to be a problem of modern man, and it's only because ancient man was full of parasites, and they were there through evolution, and they were beneficial. They counteracted something in our environment or something in our own deficit in our immune system or something of this nature.
Yeah. Well, and I had a biology professor who all the time would say a good parasite keeps its host alive. A bad parasite hurts it. That's something else to remember too, is by definition, a parasite is not trying to kill animals.
They need you more than you need them. But
there's an interesting paragraph in here saying that some estimate that up to 70% of all animal species are parasites, making parasitism the most common consumer strategy on Earth. And parasites account for a significant portion of biomass and up to 78% of food web links in any given ecosystem.
So are they just talking about children?
And they say the presence and diversity of parasites has also been suggested as an indicator of ecosystems with a low degree of human degradation. So when humans come in and disrupt the ecosystem, you lose parasite number, parasite diversity, and then you have like large dominant species or the abundant species become more abundant.
Yeah, and that's where this mention earlier of parasites being up to 10 times more vulnerable to extinction than their hosts comes in, is that they definitely they're vulnerable. And we think about wild spaces, we think about wild ecosystems and habitats. And the food web is real, guys. Everything's connected. Everything serves a purpose, no matter how small. And it's a fine web of connections. And when you remove some of those connections, sometimes the web can stand and sometimes it cannot. Sometimes those connections are so fortifying to the web that it all falls apart.
Yeah.
So this is kind of reminding us of the fact that even parasites serve a purpose in wild habitats. They've evolved to exist on this planet where they do for a reason. So,
Awesome.
speaking of gross, creepy, crawly things, let's talk about roundworms. Let's talk about C. elegans, worms, and teenage
Science's best and
angst.
favorite worm. What?
You know, like the roundworm, he's 15 years old. He's looking to move out of his mom and his dad's house. He's like, I hate you guys. And he slams his door. No?
Yeah.
I don't want to. I no.
Okay. Well,
Yes.
Yeah.
scientists at the Salk Institute were studying roundworms and humans, their brains, and the differences between adolescent brains and mature, stable adult brains. That in itself should tell you something. That they are characterizing them as mature and stable as adult brains and adolescent anything. But so looking at roundworms, roundworm brains are pretty different than human brains. Roundworms only have about 302 neurons. Humans have almost 100 billion. But there are similarities in the way our brains work. And because there are less neurons, scientists can actually study the roundworm brain easier. They can isolate specific neurons and more accurately map what's going on. So they already know the entire roundworm neuron anatomy and functionality. So from there, in experiments, they can interpret a lot more than you can from a CAT scan on a human. So they were looking at C. elegans, and they were looking at their response to stimuli, and they found that overall, the adolescent worms have different food-seeking preferences and abilities, and they are less efficient and less competent at finding food. And they call it ambivalence, the way these roundworms were acting. So what they did is they put diacetyl, which is buttered popcorn smell, and it's found in a lot of foods that we eat and a lot of foods that the roundworms eat. And they put some of this food, some food with this smell in the center of a dish, and they put a neutral odor on the other side. So they put the worms in the middle, sorry. They put the buttered popcorn and food on one side, and they put a neutral odor on the other side. Then they watched the way that the worms moved toward the buttered popcorn smell. Adolescent worms meandered, took their time. Some of them never made it to the food. Adult worms went straight for it. No nonsense. And a collaborative researcher, Laura Hale, said, it's like the younger worms are angsty teens. To watch their behavior, it's as though they say, yeah, I know I'm supposed to go over there, but I just don't feel like it. So they tested other smells that were known to be appealing to adults, and adolescents. Again, they kind of meandered around. And then when they used a repellent odor, everybody fled at the same rate, meaning the worms smelled the same. Their smelling abilities and their ability to recognize bad smells are essentially the same. So something's happening in their brain that's making them feel less motivated to go straight to the smell that they want. And this research indicates by looking at specific neuron firing in their brain and then also comparing it to studies on humans, their belief is that the neurons firing are different and they're actually kind of more scattered. So in adults, they had a very select group of neurons firing consistently, but in and more strongly, even in more subtle concentrations of the smell. But in the adolescents, the firings were kind of all over the place. They weren't as well clumped, and they were less responsive to lower concentrations. So actually, they're thinking that what's actually happening here is that the adolescent's brain is more plastic. It's less set in the way that it works and what it responds to. And so they
haven't learned what they like yet, really.
They haven't learned what they liked.
Yeah.
They haven't decided, their brain hasn't figured out how to fire in the most succinct, efficient way yet, because it's still essentially learning, which kind of reminds me about how they say that the adult brain doesn't stop growing and maturing until 25, 28, whatever research you read, right? But it's kind of based on that. Is
we say people are adults at like 18, but they're not. They're really not.
Yeah, absolutely.
And just to be fair, there's some 70-year-olds sitting around chuckling, going, "What are these kids talking about? You think they're adults? They're still growing up."
Yeah. Yes, because they know what buttered popcorn smells like, and they know when they want it.
Yeah, I would also guess that there is, there's the learning curve of the neurons responding to things. You have the neuron response. An organism discovers that the smell is associated with this food source that's awesome, and I really like it. So next time the neurons are going to fire more strongly, more efficiently. It'll get better with time. But then those that don't figure it out are going to die because they're not going to get to the food.
Yeah.
So they're going to starve. So you have a weeding of the unfit brains.
Yeah.
The adolescent brains that are just never going to develop right.
Yep. That's a very good
out.
Yep.
Survival.
Yeah. It's
what if I just don't like buttered popcorn? Like,
God,
there's gotta be something else to this life than just.
What if I don't want to follow in your steps of running to buttered popcorn?
I want
Mom
to
and
find
Dad.
something else out there. There's an whole wide world out there. It's not all about buttered popcorn. I'm going to prove it.
Yeah. So one of the other researchers said, instead of merely being rebellious, teens, both humans and worms, may just be staying flexible to adapt to an unpredictable world.
Yeah, just might be optimistic too. Yeah.
Flexible.
Yes.
Well, I hope everyone out there stays flexible while we take a very short break. And, we're going to be back in just a few moments. This is This Week in Science. Stay tuned for more fantastic science news.
An hour into what was supposed to be a quick show.
This is supposed to be a short show, you talkative people.
Whoops.
Hey, everybody. I want to say thank you to so many people for snapping up the 2017 calendars that Blair made. These calendars are amazing, and we are down to the last 4. I think we have 4 calendars left, so they may or may not be there if you wanted to get one. But if you got one, congratulations, because these calendars are fantastic. They're great. We're down to like 4 calendars left. It's awesome. Tomorrow night, SF Sketchfest, California Academy of Sciences. You can check out our Facebook page, facebook.com/thisweekinscience and go to the events to find out more about this event, or you can go to Cal Academy of Sciences website, calacademy.org, and look for their nightlife page to be able to find out about the SF Sketchfest event that we are at tomorrow night, going live at 8:15 p.m.
Maybe you can come and buy one of the 4 left calendars, and we'll sign it for you.
Calendars. It's amazing. Yes. TWIS also has merchandise that you might enjoy. Head on over to twis.org to get yourself some of our very quality swag. Not low quality swag, but great quality swag. Our merchandise. We have got at our Zazzle store Lots and lots of fun things. Hats, mugs, t-shirts, polo shirts. Blair put together a polo shirt for everybody. So again, if you just go to twis.org and click on our Zazzle store link on the main page, that will take you to our Zazzle store, where you can find all of these neat products with the TWIS logo all over them, even a mammoth pillow or TWIS bullfrog wrapping paper. Right? I mean,
or a dinosaur or something. And a dinosaur phone cover.
A dinosaur phone cover. Thank you, Kai.
And a turtle belt. And then a turtle wrapping paper. You can get turtle wrapping paper.
Mm-hmm.
Yeah. What else do you see there? What does that look like? Another dinosaur thing? Oh, I like the mammoth pillow.
Yeah.
It looks pretty cool, doesn't it? Oh, no. Pick it up. Hey, no, you can't have it. No, I need to finish what I'm doing.
Yeah,
Kai.
more images from this year's calendar will be coming to the Zazzle store soon. I'm experimenting with some new items like the pillows. So if you buy some, then we'll know what you like, or you can tweet at me or send me an email and make a request. Maybe we can make something that you're interested in. So let me know.
Yeah, that would be fantastic. Additionally, if you go also to twis.org, looking at, there's all sorts of things. There's a tortoise tie.
Yeah.
Kai, you like the tortoise tie?
Yeah. Wait.
What's that? No. Kai wants to sell you some things.
Yes.
Kai, what do you think people should like?
I think people would start to like, I think that mouse phone cover.
Mouse phone cover?
Yeah. That'd be really nice. Wait, what is that?
A jigsaw puzzle, a T-Rex jigsaw puzzle. You can have a T-Rex plate, or you can have rat playing cards, or a T-Rex clock.
That'd be pretty awesome.
That'd be pretty awesome, yeah.
You can get mouse collars.
You can get mouse, yeah.
Rat collars.
Little mouse collars for your a rat for your cat.
Yes.
Or your dog.
Or your dog.
Did I see a mastodon throw pillow?
Yes, you did.
I really need that. I really need that.
There's you can have a little baby onesie, rat onesie. Look at all these cool things.
What's a onesie?
It's for babies. It's a one piece. See how it goes under the. Yeah. Okay. Kai, you're sleeping. Oh yeah. Also, if you go to twis.org, this is where we also connect with you for support in the monetary fashion. So if you are interested, you can click on our donate button that's on the main page, Donate to TWIS. It's a nice button. You can donate using the PayPal interface, super easy. Or up in the main header bar, there's a link for Patreon if you're interested in becoming a supporter through the Patreon interface. This is another great place to go. And super easy. Twis.org, it's where you go to find all the different ways that you can interact with us and become more a part of the community, a part of our minion family, right? If you can't afford a donation, however, look at our run sheet. Here, I'm screen sharing the run sheet. There we go. If you are unable to afford a donation, we can always use your help. So if you are able to share us on social networks like Twitter or Facebook or Snapchat or Instagram, if you get a new calendar in, take a picture of yourself, Snapchat it, Instagram it, tell people about it. It's kind of awesome.
Or tell a person in person that there's this digital thing that they can have access to. That's entertaining.
You can also share the podcast from the iTunes podcast app. You can send it via email or messenger or text message, and then it kind of pokes your friends to download it themselves.
We thank you for your support. We really could not do this without you.
What is the moral that we all must learn, especially those of us with money to burn? Before your eyes widen at the book on the shelf, think. Is he helping you, or is he helping himself? You can't believe what a skeptic I am. You should believe what a skeptic.
And we're back with more This Week in Science.
Oh, yeah. You got a pet rock for me, Justin?
So, yeah, this is a great story. What separates us from Neanderthals? We shared so many characteristics back then, it's hard to say we were much different at all. Perhaps we remain truly, even to this day, very similar to our Neanderthals. Because it turns out one of us used to have a pet. And as Kiki alluded to, that pet was not your typical modern pet. Though in the mid-'80s, there was a fad that swept America for decorated pet rocks. Some stores even sold them, fuzzy hair and googly eyes already glued on. You could buy a rock with googly eyes on it. Yeah, they would have them like up at the counter. You'd be like punk rock and all rock and
Punk
anyway.
rock.
But now and international. No, this is a true thing. You guys don't remember this? I remember.
No, I
this.
know.
But
I'm laughing at the
Okay.
pun of the pet rock. Punk rock.
An international group of sciencey researcher type people has discovered a brownish piece of split limestone in a site in Croatia that suggests Neanderthals 130,000 years ago collected the rock. If we were walking up and picked up this rock, we would have taken it home, says David Freyer, a professor emeritus in anthropology who was part of the study. It's an interesting rock, he says. Exclamation point was mine. Finding is important, he said, because it adds to other recent evidence that Neanderthals were capable on their own of incorporating symbolic objects into their culture. This rock was collected about 100 years ago from a Krapina Neanderthal site, which is items preserved in the Natural History Museum in Zagreb, Croatia, where in recent years, research team has had time and access to re-examine them. Davorak Radovic, created in the Croatian Natural History Museum, was the study's lead author, and Freyer was the corresponding author. Same research group was widely recognized back in 2015, covered on this show, when they published in PLOS One about a set of eagle talons from this same Neanderthal site that included cut marks and were fashioned into a piece of jewelry. So this site, ancient as it be, shows artistic leanings or cultural iconography of some sort amongst these folks. But the cave where this was found, the bit of is a site with sandstone. So that this is limestone rock is not from this cave. In fact, they find that it was from several kilometers away. Possibly there's other sites in the area. So this rock had to be found somewhere, picked up, brought back to the cave, and there was no chipping on it. They weren't cutting with it. They weren't smashing it. They weren't fashioning it. They apparently just liked the look of this rock.
Cool.
Yeah. It says the fact that it wasn't modified, to us, it meant that it was brought there for the purpose other than being used as a tool, Freyer says. Look at the rock also caught researchers' eyes. Many inclusions of black lines on it stood out from the brown limestone. Maybe this, they think, is why it caught the Neanderthal's eye and it took it home. So yeah, Neanderthals, aside from all the other wonderful things we've been learning about them, they could appreciate a good-looking rock.
They like trinkets.
Just for its symbolic capacity of being
like an
interesting.
octopus tending to its garden. The Neanderthal collects its fancy rocks. Right?
Yeah, I think the only thing that could have made this story a little bit better is if that rock had a fossil in it.
Yeah.
And then then it was like, oh, Neander anthropology would have been born. But yeah, just a neat looking rock.
Just a cool rock the Neanderthals liked.
But this is also 130,000 years ago. I mean, early that year, old ago-ish, humans were sort of discovering interesting artifacts and starting to collect things. But it seems like the Neanderthals were just a bit more advanced earlier in that area.
Go Neanderthals. We learn more and more about them and how much, I mean, really that's the interesting part of it, is how much more advanced they were as we get more and more information than we've ever given them credit for.
Yeah, well, I mean, we didn't have anything to credit them with initially, too. But
Yeah.
there was a very large sort of, we're modern human, and so anything older than us was brutish by nature. Had to be. I mean, and I guess you can excuse it too, as not just sort of
number one. We are number one.
Yeah, I think we can excuse away a little of that too, is that we were also looking, as Neanderthals were being discovered, we were looking for a link between man and ape. So our assumption, lineage-wise, may have been to make it more ape-like or brutish or animal-ish, in our early concept of the time frames of evolution. But now we understand that there was a much longer development and experiment that led to mankind. And this, even 130,000 years ago, ain't that long ago, and the journey that we've taken.
No. Well, keeping to other animals, it's not, we've entered this wondering how animals do things, right? How do we move forward? Let's move forward to killer whales.
Mm-hmm.
Rock collecting Neanderthals to killer whales. Did you know in the world, animals that we know of, only 3 go through menopause? Do you know which 3 animals go through menopause?
Well, since you said you had a killer whale story, I guess that's one of them.
And then the other one's humans.
I'm fairly certain humans.
And then
third.
And the third is short-finned pilot whales. So 2 species of whales.
What?
And 1 species human.
I would have gone ape.
No apes.
Oh boy.
Yeah.
Go through menopause. And so
Holy
there's been a huge question. Why do humans go through menopause? Why do women lose their childbearing capacity? Well, why do killer whales lose their childbearing capacity? Why do these short-finned pilot whales do the same? What is going on to lead females of these 3 species of the entire animal kingdom that we're aware of to stop bearing children? So, what a new University of Stirling out of the United Kingdom, and also University of Exeter in the United Kingdom, looking at killer whale populations. They found that, so they know that killer whales live a really long time after menopause. But then we also have elephants who don't go through menopause and also live for a very long time.
Mm-hmm.
So what's going on? They looked at 43 years of data on 2 Pacific Northwest populations of orcas. And in orca families, the sons leave the pod. So the families, the social groups are known as pods. And there can be many related and unrelated individuals within a pod. However, males tend to leave the pod and go find other females in other pods to mate with. And males die at around 30 years old. The mothers and the daughters, they stick together and they live together. And they all usually live for long, long time, although the females stop producing when they're around 30 or 40. Yeah, so killer whales actually can live about as long as humans.
So were killer whales the subjects of the stories that we've talked about why grandmas exist?
Maybe.
That sounds familiar.
Yeah, possibly. So yes, and this is what happens. The mothers, that's part of this story, is the mothers stop having children. Their daughters still live with them. Their daughters enter reproductive age, and the mothers become grandmothers and help to rear the daughter's offspring, similar to what happens often in human society as well. So what they looked at were births of different, throughout these 43 years of data collection at the births, what calves were born. So 161 out of 525 calves were co-generation births, and about 31% of the babies died. And in these cases, according to the Science News article, the older mother was most likely to lose her calf before it reached 15 years of age. And the mortality of older mothers' offspring was 1.7 times that of younger mothers. And so what the researchers report in their current biology paper is that it's this cost that you've already had one offspring and your daughter is starting to go into reproductive age and maybe has a baby. And if you have a baby too, you're basically going to be having your own baby with no one helping you to take care of it and potentially helping out your daughter as well. So the grandmother is taking care of 2 generations below her. And it's this high cost, which may have driven the evolution of menopause. Because if you're 1.7 times, if the mortality of your offspring is 1.7 times greater, maybe that's enough to push you into the realm of just not having offspring anymore. The cost is too great. It's not worth it to you. It's not, you could die in childbirth.
Whereas if you help out your daughter, those are still your genes that you're helping.
Still your genes, exactly. Yeah, so it's the, another researcher in the article says that he thinks it's Richard Connor from University of Massachusetts in Dartmouth says he thinks it's a cool study because we usually think about these kinship issues in terms of helping behavior, not competition. And so what's going on here in reality is that, you know, there's competition for food, for resources also. And so the daughter and the mother are trying to provide for their offspring at the same time. And the daughter is probably more likely to get food for her offspring than an older mother also. So
it's just a combination of all of these factors that lead to menopause making sense. It seems like we see some of these factors in a lot of animals. We see long life. We see reduced resources. We see family units. But having all of those things kind of in one big perfect storm means it makes sense to have grandma help out and not reproduce anymore.
Right. And if grandma can help out by, you know, going and showing her daughter where there's a bunch of salmon or helping them find a lot of salmon for them to eat, then the baby's going to do a little bit better. And the researcher on this paper says, what's his name? Darren Croft says that human families we're not exactly like killer whales, you know, where it's like the sons go off and the daughters stick around with the moms. But our society, our social groups are organized around the provisioning and sharing of food. And so maybe there is something to the cooperation, the sharing of food, the resource provisioning that has led to reducing conflict because of reducing competition and increasing the likelihood of menopause. So maybe human menopause has something to do with it, the same story that killer whale menopause does. But find it fascinating. Humans, killer whales, and pilot whales, short-finned pilot whales. Menopause. Nobody else.
Yeah, that's wild.
Who knew? Yeah. What's up, Justin? Give me another story.
Oh, is it that time again where I go on about persistent infection? People often get sick from a type of illness that they're then protected from if they, are introduced to it again or it's introduced to their system again. Some of these infections, like chickenpox, small number of microbes persist in the body a long time after the symptoms have gone away and the infectious part of the infection has gone away. These microbes can reactivate is waned with age or illness, and the disease can resurface. So most of our attempts to treat these sorts of things are based on complete sterilization and eradication. And in reminding me sort of somewhat of the parasite story, researchers are now looking at this, and they believe that it might actually not be a good thing to completely remove an infection. Because persistent infections, those that have small amounts of microbes that still stick around for a really long time, even though they carry a risk of reinfection with the waning and the aging, also keep the immune system primed to prevent another infection. What are you laughing at?
That seems crazy to me. Because they tell you over and over and over and over and over again, take your complete course of antibiotics. Don't leave any survivors.
Whoa. We're not suggesting to do something other than that.
Okay. I think you need to clarify.
I'm clarifying right now. Yeah, it's not necessarily that. So these are,
because you just told me to leave an infection.
Yeah. So here's what they found.
Persistent infections, though, those are ones that are they're in there, and
you don't normally get rid of it with an antibiotic.
You can't get rid of it with an antibiotic. Yeah, it just sticks around no matter what.
So they were looking at leishmania. I'm unfamiliar with this disease.
Leishmania. Mm-hmm.
It's a group of parasites that cause ulcers on the skin, can infect internal organs. As estimated 250 million people worldwide are infected with this parasite. It's only really found in tropical areas. 12 million have active disease. So 12 million people are in critical suffering. There's another 250 million people who are infected. The disease can be disfiguring, even fatal. Once a person is infected, he or she is protected from getting sick a second time. So, long-term infection confers with it long-term immunity. You got infected with it, you survived the initial infection, and now you're kind of immune to it. You're not going to get it a second time. Unless it's completely eradicated from your system. In which case, you can get the really critically ill portion of this disease again. So people are, they harbor the parasite at low numbers for years until they recover. The persistence may lead to, so they did a study in mice that showed that completely clearing the parasite can make the animal susceptible to another bout, going back around, getting the infection all over again. Studying mice, researchers used fluorescent markers to distinguish different types of mouse cells and found that most of the parasites live in immune cells. The parasite lives in an immune cell that is not only an immune cell, it is an immune cell capable of killing the parasite. Yet, despite this, sort of wrong place to house yourself, the parasites appeared normal. They were normal shape, size. Furthermore, the parasites continued to multiply, yet the total number of parasites stayed the same as time went on. They continue to multiply. The numbers remain the same. This is one of the researchers called it the Jimmy Hoffa effect. They couldn't locate the body. We were unable to show directly the parasites were being killed, but some of them must have been dying because the numbers didn't go up, even though multiplying was still taking place. The immune cells that house the parasites are responsible for killing those very pathogens and activating a more robust immune response. In this process, the ongoing multiplication and killing of parasites that the researchers believe underlies the long-term immunity associated with persistent infection. This explains why people typically can't get sick from the same pathogen twice. But it seems as though the immune system was almost like parasite husbandrying this infection. Like, we're going to keep a sample. We're going to keep it limited to this size. And as long as we've got it, we can spar with it just enough to be on the lookout for, it was sort of like its identification marker. Like, this is what we're looking for. If you see this outside, You go out there and kill it. These ones, they're okay. It's like a little pathogen zoo that we've created here so that we can get up close and friendly. You can look at it through the glass. But if you see this in the real world, run or fight. You got a couple of choices. So usually scientists design vaccines to get sterilizing immunity, trying to kill all the bugs, says Beverly, one of the researchers involved. But what you really need is protection against the pathologic consequence of the disease, not necessarily sterilizing immunity. For some of these organisms, solid long-term protection may come at the price of persistent infection. And as we were sort of talking about, like, I almost felt for a moment that we had the same story. This parasite is a microbial parasite. This is an infection story. But it seemed to really kind of fit, almost theme verbatim, what your parasite story was earlier, because they're part of the ecosystem. It's part of the immunology, the immune system's ecosystem, to have these microbes present so that they can be more robust in defending against them in the future. Right.
They serve as like training weights to help them be strong enough to fight.
Kind of. Also, don't do that. Don't do training weights. Don't tie sandbags to your ankles. It's really bad for your knees.
Okay.
Yeah,
ruin my metaphor.
I mean,
take all your antibiotics.
Yeah, I mean, there's a huge question there. Do you get infected? I mean, if you get infected with something, do you keep the infection?
Well, and here's
get rid of the infection,
and
on the chance that, you might get it again and it'll be worse.
Yeah, and here's where I think you can draw that line. This is the leishmania or
causes leishmaniasis. Yeah,
leishmaniasis. If you contracted leishmaniasis while visiting the tropics, probably safe to just completely eradicate it from your system. Yes. But if you caught it and you are in the native area where this is persistent, maybe you don't totally clear it. Maybe the benefit of your immune system being ready for another encounter outweighs getting rid of it altogether. And so it's going to, I think that's sort of the dividing line between which strategy you use. Is this something you're likely to see again, or is this something that it was like a one-off vacation mishap?
Yeah. Then there are others. So like in the beginning, you mentioned, so there are lots of microbes that can cause these persistent infections. And so like herpes virus is one, chicken pox is another, and when you get rid of chicken pox as a child, there's the possibility, or you get chicken pox as a child, there's the possibility of getting shingles as an adult. And so there are these, different sides to the infection, and how your body will react to it. Yep. Fascinating.
So I didn't even think of herpes. So herpes. Now it prevents you from getting herpes.
Herpes prevents you from getting more herpes.
I don't know if that's actually true, so don't quote me on that.
Yeah.
But if you already have herpes, the chances you contracting herpes is you already have. Yeah, I guess that's immunity of a sort.
It does affect your immune system for sure, long term. Yeah, interesting questions. These are interesting questions. So let's talk about eels for a second before we come to the end of the show.
Yes, we can't let this show go by without discussing it, at least for a moment.
Let's get real about eels.
Okay. Let's get real
about eels. So European eels have been discovered to migrate, and European eels usually, they're found in rivers and creeks. At least they used to be found in rivers and creeks in great abundance. Same thing here. We've got the sucker fish up the Columbia River. What are those called? Like, anyway, they're good food, but they only found adults and they never found babies. So what do these eels do? What do these real eels do to go mate and have babies. And so it was discovered in 1922, a Danish biologist called Johannes Schmidt. He discovered that there is the Sargasso Sea. It was a massive swirling gyre in the North Atlantic Ocean.
Wait, what's a gyre?
Gyre.
I have no idea what that is.
Well, it's just a big, slowly spinning
Whirlpool
area. Well, yeah, but you think of a whirlpool as being like super fast spinning, like in your drain as water goes down. But this is just more of a rotation point.
A gyre. Gyre. Sorry.
Gyre in the North Atlantic, the Sargasso Sea, and discovered that, ta-da, this is from Europe to the Sargasso Sea. This is where these eels have been going. They journey to the west to the Sargasso Sea, and they mate, and they hatch their eggs, and then the Gulf Stream carries the eggs back, and 2 or 3 years later, ta-da, more eels in the rivers and streams.
Thank you.
And so they thought, yes, one migration, kind of like salmon. You spawn, you go, you grow up, you're birthed in a place, and you go out to ocean, and then you come back to spawn, that kind of a thing. They thought it was just a single, easy migration, but there's there wasn't really any evidence to support it. And so recently, some researchers got together, and this is pretty cool. They've been looking at eels, tagging them, and they've got projects that are called the Codicy and the Iliad.
Oh my gosh.
Talk about funny puns.
Yes.
But they finally had tags that they could use to tag eels to find out where they went and when they went and what happened. And so they tagged more than 700 large female eels. And if you think of eels just as those long, wonderful, fishy fish that they are, they're also slimy. So could you imagine?
Difficult to catch.
Yeah, they're slimy. Ooh, a little slippery. Slip out of your hands. So not easy to tag them, right? Yeah. Anyway, over 700 were tagged. And then they tracked them. So these tags logged environmental data, and then when a tag's battery died, it detached from the eels, because the eels swim like 200 meters under the water. They swim very, very deep. And so the tags detach and go to the surface and go beep, 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 and relay their GPS information. So by satellite, or end up getting drifting back to shore for collection, and then their GPS data can be taken off of them, so that, ta-da, you have a track of where these eels have gone.
Sure.
And so they discovered that not very many eels actually made it out of the rivers to the ocean. So out of over 700 tagged eels, 87 tags were collected beyond the coast. 87.
Huh.
So less, around a 12th. And then a lot of those got predated once they got into the ocean, and none of them made it past the Azores. So what they're finding is that these eels are taking a really long route. They're not just like super migration, let's go and swim really fast and spawn and get it all done. They're kind of taking their time. Like, there's some fast swimming eels, but most of them are kind of slow. They're taking their time, and they're not doing it in like a one season go round. So they're thinking that a lot of the eels don't actually make it to the first spawning season after they leave the rivers and streams, and that maybe it takes 2 or 3 seasons for them to even get to the Sargasso Sea. Yeah. So these eels that they thought were just like
keep gyrating around.
Yeah. They thought that, hey, they're just going back and forth super fast and doing their spawning and blah. Nope. No, they're not. They are not. And so the researchers propose a hypothesis that maybe there's a mixed strategy, that some of them go quickly and some go slowly. And so there's potentially different eels with different, I guess, excitement about breeding, whether or not they want to get there quickly or slowly. And so there's the alternative of the eels that leave too late or don't go very, very fast might just not succeed in spawning. All right. Anyway, eels migrating. And nobody really knows anything about them.
We know a little bit more now.
We know very slightly more about them, but it actually, it's funny. It's like, okay, 1922, hey, we found these eels in the Sargasso Sea. Let's make up this story about the eels and their breeding life cycle. And we really don't have a lot of evidence for it at all. And now we finally have evidence, and it kind of makes the picture a bit more confusing.
Yeah.
Than it was.
It raises more questions.
It raises more questions.
Guess what? More research projects, folks.
Yeah, well, I mean, if anything, I guess what we might learn from it is that it's not. Wow, there's a bit of chaos to the breeding.
A bit of chaos, yep.
which you don't expect to see in something that's evolved and been around for a really long time. You expect to be able to track down the parts of it and have nothing relied on chance or ocean current or sort of random placements. But yeah, to all of us, there's a little bit of chaos that leads to our eventual result.
Wow. A little bit of chaos in the eel world. The eel world. Blair, tell me about spider silk. I talked about spider silk. What else could you possibly have to add?
Oh my gosh, just you wait.
Spider silk.
Just you wait. 5 years work in an interdisciplinary team of scientists at the University of Nottingham has found that they can make antibiotic synthetic spider silk. What?
Oh, no.
Antibiotic synthetic spider silk. So chosen molecules can be clicked into place in soluble silk proteins before it's been turned into fibers, or even after the fibers have been formed. And so the process can be easily controlled, and more than one type of molecule can be used to, quote, decorate individual silk strands. My favorite part of the story is how it all came together. It was at a discipline bridging university sandpit meeting 5 years ago, and a chance meeting between professors provided to be the start of this 5-year project. Dr. Goodacre, who heads up the spider lab in the School of Life Sciences, got up at the meeting and showed the audience a picture of some spider silk and said, I want to understand how this silk works and then make some. And then another professor came up to him and said, I think my group could make that. And suggested some interesting tweaks. And now they've been able to adjust the silk proteins in all these different ways. And that includes antibiotic. The really great application of this is that spider silk actually used to be used in medicine a long time ago. Back in the Civil War era, they actually would wad up spider web and try to close wounds with it. And actually, there's a lot of opportunity to use spider silk for that.
Yeah, for wound dressing. Yeah, especially
for wound dressing. Absolutely. And also for tissue regeneration. It's actually, I think it can serve as a scaffold for cell growth. So all of those things with a kind of time-released antibiotic component could be a huge boon to the medical field. And it's all from antibiotic spider webs.
That's cool. So those people need to talk to the people that I talked with last week, talked about last week, and suddenly we'll have kilometer-long antibiotic spider silk.
Yep. Indeed. So they need another interdisciplinary sand pit.
Put it all together.
It's also the best material for creating an elevator to space.
If they could actually get it to work. But no. Spider silk? I don't think so.
I don't know.
Oh, space elevator. We haven't talked about the space elevator in a long time. Do people even talk about that anymore?
Yeah, that one's kind of gone away for a while. Yeah.
Let's find out.
You mean like Willy Wonka?
No. We'll do it.
Kind
We'll
of like that, only with hot air balloons and a really long cable that then creates a launch platform in low orbit, which requires less fuel, and so it's the whole thing.
That sounds like more trouble than it's worth. Have we done it?
No hot air balloons.
Is it time to close out the show?
We're a bunch of bags of hot air. It's time to close out the show. It absolutely is. I would like to say thank you to everyone who's watching. Thank you for watching. Thanks for those of you in the chat room for chatting. And thanks to our Patreon sponsors. Thank you to Chris Clark, Paul Disney, G. Burton Lattimore, John Ratnaswamy, Richard Onemus, Byron Lee, EO, Jared Lysette, Kevin Parachan, Andy Groh, Keith Gorsell, Jake Jones, Gerald Sorrells, Ulysses Adkins, Derek Nickel, Dave Friedel, James Randall, Eric Schwald, Bob Calder, Mark Mazzaros, Ed Dyer, Trainer 84, Brian Hedrick, Layla Marshall Clark, Charlene Henry, Larry Garcia, Randy Mazuka, Tony Steele, Gerald Onyago, Steve DeBell, XV, Daryl Lambert, Haroon Sarang, Melissa Mosley, Alex Wilson, Jason Schneiderman, Greg Guthman, Dave Neighbor, Jason Dozier, Matthew Litwin, Eric Knapp, Jason Roberts, Richard Porter, Rodney, David Wiley, Robert Aston, Aurora Lee, Bill Kersey, Ben Rothig, Darwin Hannon, Rudy Garcia, Nick Abassi, Felix Alvarez, Cosmic Gypsy, Brian Hone, Orly Radio, Brian Condren, Mark, Nathan Greco, Hexator, Deborah Smith, Much Neves, Flying Out, John Crocker, Christopher Dreyer, Sylvan Westby, Artyom Shuwada, Dave Wilkinson, Steve Mashinsky, Rich Gramus, Gary Swinsberg, Phil Nadeau, Braxton Howard, Salgood Sam, Matt Sutter, Emma Grenier, Philip Shane, James Dobson, Kurt Larson, Stefan Insom, Michael George, Russell Jensen, Mountain Sloth, Jim Drapeau, John Maloney, Jason Olds, James Paul West, Alec Doty, Luma Lama, Joe Wheeler, Dougal Campbell, Craig Porter, Adam Mishkon, Aaron Luthen, Marjorie, Paul Statton, David Simmerly, Taylor Harrison, and Columbo Ahmed. Thank you for all your support on Patreon. And if you're interested in supporting us, you can find information at patreon.com/thisweekinscience. Also remember that you can help out simply by telling a friend about TWIS. On next week's show, once again, we'll be broadcasting live online at 8 p.m. Pacific time on twist.org/live, where you can watch and join our chat room. But don't worry if you can't make it. You can find our past episodes at twist.org/YouTube and twist.org. And we are going to be having an interview about nothing.
Huh?
So it's me? It's going to be me on the show?
No. Oh no.
Justin. It'll be Justin.
We're gonna be talking about nothing. The meaning of nothing.
Oh.
Nothing in physics. What is nothing?
My head hurts already.
We're gonna be
So we're talking about Blair's love life and my finances?
The void.
Go.
Nothing.
But
and don't forget tomorrow night, Cal Academy of Sciences. January 19th.
When is that?
Tomorrow night.
That's going to be tomorrow.
San Francisco, California Academy of Sciences, Nightlife, SF Sketchfest. We go live at 8:15. Doors open at 6 p.m. You can find information at our Facebook page, facebook.com/thisweekinscience.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have a mobile type device, you can Google TWIS for Droid in your Android marketplace thingy, or simply This Week in Science in anything Apple Marketplace.
If you are dying to know more information about anything that you may have heard here today, show notes will be available on our website. That's at www.twis.org.
What's that website?
www.twis.org, like TWIS, like This Week in Science. www.twis.org.
ORG?
Yes, for organization, where you can also make comments and start conversation with the hosts and other listeners.
Or you can just contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, that's, what was it, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here tomorrow, actually. Actually, I'll be in front of the flamboyant cuttlefish in the Calorie County. But we're going to do some science too tomorrow. And then we'll be back here next week.
Yes.
And we hope to see you in both instances once again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
And that's the end of our show. The 1 hour a week show that we just finished. Yes. Now it's the post show.
Oh no.
All right, so you are going to be able to play music
Oh, yes.
tomorrow.
Let me make sure I have it on my telephone.
Okay.
I know I have it on my computer.
Okay. That's good because we can. I don't think we need it on your phone. If we have it on your phone, that's a good backup. I'll also bring it on.
Oh yes,
flash drive for the peoples. But
have it.
Ed quit. What?
I have this. No, I have it all. All right. Great.
That was a good show. Longer than I expected it was going to be.
Yeah.
Just getting like the talking, and everyone's like, "Oh, we'll just talk about the story. Let's keep talking about it."
But sometimes it's interesting.
It is interesting. I know. Some interesting stuff out there. So tomorrow, where's Justin?
Oh boy. The everlasting question.
Ed, PC is much easier typing. Yep.
So, okay, so I'm trying to see.
What picture on Twitter? Dale Poco. Oh, this picture? That picture on Twitter?
Thanks, Identity 4.
I don't think I'm in a Women of Science picture.
Identity 4 says, I like it when you guys talk about science stories. I don't like it when you don't talk about science stories.
That's perfect. Perfect. Oh my goodness. So I'm tired. I still have to pack. My child fell asleep though. Look at that. He finally stopped wiggling around.
He was wiggling. He was like a C. elegans.
He has so much movement in him. And it's
but
so hard.
Not moving towards the buttered popcorn, unfortunately.
Yeah. 10 other scientists.
Ooh, we have some scary mouse stories tomorrow.
That is good feedback, right? Dictel. What? Mouse story? What?
We both have scary mouse stories.
Oh, do we? Are they different stories?
They're very different. Yours is about eating,
secret,
and mine is about sex.
Awesome.
And I don't have anything for tomorrow yet, but I plan on finding something between now and then. But I got a hard out. Because I gotta finish working on a thing.
Okay, so let me tell you before you go. We're gonna have to keep to time tomorrow night because we only have 40 minutes for our show.
I'm gonna bring 2 stories.
Yes, you bring 2 stories.
I'm bringing 3, but I'll drop one beforehand if we want.
Yeah, I have stories I can drop. Not that I want to drop, but that I can drop. And then I have a number of stories for the end of the show that are basically just going to be like little blurbs. But I can drop them if I have to. I don't need to have them in there. I'm excited about one of my stories, though. I'm excited about a lot of my stories, actually.
I'm really excited about my quick story for the end of the show. I have to get more excited about the other stories I brought or find a new one.
Yeah. That happens
around. I'll check tomorrow morning, make sure there's nothing breaking. And if there isn't, then I'll dive a little deeper into my stuff.
I'm doing this story because I feel like I have to. But I don't like it. And then I find something last minute. I'm like, I like this better.
Yeah. I love this one. Identity 4, you're hilarious. That's right. Justin has to wear a muzzle. No, I'm going to have a timer. I'm going to use my phone.
Wait a sec. Identity 4. We've gone down and we've broken down minutes of the show before. And it has scientifically been sussed out.
That you talk way more than both of us combined.
That I actually utilize less of the time that we are on air than my counterparts.
This
combined.
Does not sound like science.
This is true. We did this.
No, we have not.
We did a whole breakdown of the minutes of time that were being used on the air for this, that, and the other story. and I was keeping the time.
No. Stories, sure. The question is time on the clock, your voice versus Kiki's voice versus my voice. That's the question.
You guys spend more time talking than I do.
No, I will not believe that.
It's absolutely true.
But I will say we did fine in Baltimore with the timers. We did really good.
Timers
The
help.
timer was cool.
Yeah, it worked really, really well because I could also glance over and I would have a question I would want to ask, and I would see we had 30 seconds left. I was like, well, I'm not asking that question. Or I'd see we had like 3 minutes, so I could ask the question. And it was nice because I didn't feel anxious about slowing things down because I could see where we were.
Yeah, no, I think it'll be handy to have a timer running.
Yes.
So, but I gotta go finish this thing. I'm gonna share this. I'm digging this thing. I'm working on hand.
Ooh, 8:15 showtime, Kiki. Oh,
We're in the terrace, and we have 8:15 p.m. showtime. So if you know how Wi-Fi works there, let me know, because that's where we're going to be. 8:15 p.m. in the terrace, and cool. Oak Tech, looking forward to you coming. That'll be great.
yes.
The
Are you
earthquake
from
experience.
Oaktown? Oak Tech?
Quite the
Oh.
question. So Justin, you got to go. I know you muted yourself.
Yeah, you have to unmute yourself, friend.
You muted yourself. You can't just do that. You muted yourself.
I was saying I interrupt more just because I'm trying to get a word in edgewise.
Oh, right. Do I need to pull up that study again where people thought that men were speaking more in a meeting, or that men were speaking equal time to women and they were speaking 80% of the time?
Right.
And then when women and men were speaking equally, everyone thought the women were speaking 70% of the time.
I'm telling you, we went through the numbers at one point, and it turned out I was not as
It sounds like the numbers were based on stories, not on the amount of time you were speaking.
No. Okay.
So for everyone out there, just for information, if you're watching right now and you're wondering if you can watch our show at the Cal Academy tomorrow when we go on, we are going to try. We do not know. We're going to try to live stream. But I do not know if we will be able to make it work. We will not have an Ethernet connection, so it will depend on the Wi-Fi we have and the devices that we have to be able to do it. So we are going to try to hack something together, and if we can, if I can have a Hangout running in the background of my, my computer as I'm have the run sheet up, then great. Then we do it. And we can do it from my laptop maybe, or, maybe we do it from a phone. Maybe we Periscope. Maybe we Facebook Live. I don't know how we're going to.
We don't have any idea.
No,
chances are
I will tweet. I will Facebook message. I will try and get the message out as best I can. If any of you pick up the message as to whether or not the live streaming is happening, please share the information so that other people will know as well. That would be very helpful.
If it becomes a lost episode.
But I will be, but this device here, is getting a clean audio feed of the show. Nice. So even if you do not have video, there will be an audio version of the show. It'll just come out a little bit later.
Nice. Yeah.
There will be no lost episode.
Yeah.
No.
Knock on all the wood.
My big tricorder.
Oh yes, Rob says just run the Ethernet cable from my house. Yeah, that should work.
That'll work.
We only need about 4 miles, I think.
But how far can you possibly be from high-speed internet anywhere in San Francisco at any given time? Really?
Vitriol, you're going to be on a bus late at night. Well, if you're able to watch and we're streaming, I hope you can have some enjoyable bus time.
All right. I got a heart out again. Good night, minions. Good night, Kiki. Good night, Blair.
Good night.
Talk to us tomorrow if you have any issues or anything, Justin.
I don't anticipate anything.
Okay, but you're going to be here between 2:30 and 3?
Okay, so the only issue.
Oh, no.
In Canada. Whoa. Oh, Canada.
The only thing that's going to slow my progress is I'm going to be at the mercy of the 80 and the weather. The freeway speed will determine. Normally, I can make the drive from Davis to San Francisco in about 35 minutes.
No, you cannot.
Well, I mean, given that there's no traffic.
I've made that
but
drive
that's
so
rare.
many times. The fastest I've ever been able to make it is an hour and 15 minutes.
Oh, no,
Door
I've
to
got
door.
that blown out.
Door
of
to
the water.
door from
I
in
think-
the
I think
interior Noe Valley.
I think last time I
To
went down there, I
Canius.
made it from here. Actually, it was more than 35. It was like 45 minutes, right, Blair? I was like, I'm leaving. And then it was like 45 minutes later, I was like,
Really?
I'm here. Yeah. But you know, it's going to be rainy. There could be traffic
leave
delays.
at
Oh.
12:30.
I'll be leaving slightly before about that time. Yeah.
12:30?
Yeah, actually,
Okay.
that's the
think
plan.
12:30
Maybe a little
would be-
earlier than that.
Mhm.
Yeah.
Great.
So it should be noted.
Fantastic.
Should be.
Great.
Fingers
Good night
crossed
again.
there's no
I'll
flight
see you,
issues for me.
I'll see y'all tomorrow. And the where are we going again?
The
What's
Cal
the? I mean, I
Academy?
know I'm where I'm meeting you up, but where is the?
It's the California
the-
Academy of Sciences in Golden Gate Park.
Oh, we haven't been there for years.
I know. Is this the
That'd
first
be
time you
fun.
put that together?
Huh?
Never mind.
I don't like pay attention to all the detail stuff. That's
Like where we're
too
going,
much.
what we're
Yeah,
doing.
my whole focus is I got to meet up. I got to meet up on Blair with Blair, and then Blair will drag me to where I'm supposed to actually be.
Yeah, well, luckily,
that's-
all
that's
three of
all.
us will be together at 2:30.
Yeah.
Yep.
All right.
Fingers
Good.
Good night. I'll
2:30.
see you.
Good night, Justin. We'll see you tomorrow.
Good night.
He better have disclaimer and 2 stories. Kick his butt.
Well, you can always write the disclaimer right before. The stories he can't do right before.
I mean, even that. I mean, it's a live show. You want to have a decent disclaimer, not like
Yeah.
off the top of my head.
Well, I mean, the one in Baltimore was good, and he wrote that right before.
It's true.
He's
It's
full of
true.
surprises, that guy.
Yes, I will agree. I will agree with that. Surprise, surprise, surprise. So you remember Josiah Zahner?
Hmm.
He is DIY CRISPRing himself.
Oh boy. Why am I not surprised?
Oh my god. So
I feel like he has some confounding variables in his body at this point.
I'm going to show a video that he made. So he has I'm going to screen share, and the great thing is there's not really any audio other than music that goes along with this video. But he has his blog. So if you go to ifyoudon'tknow.com, but his blog is basically josiahzahner.com. And so he's created a video for Homo sapien mutagensis. Cracking me up, Josiah.
Put it on his head. What is he doing?
He is putting different formulations. He's doing genetic engineering experiments on himself.
What?
That's a tattoo gun, a tattoo pen.
How can he do multiple experiments on one arm?
I don't know. He separated them by pen circles. He has a messy garage. Oh, you missed him curse right there. Oh, there's his cat. That's it.
Wow, what? Oh my. I don't know.
What I posted was I on his Facebook post I wrote, "Wow, wow, wow, wow. I'm going to make some popcorn." I'm just going to go make some popcorn. I'm going to sit here. I'm going to eat some popcorn. I'm just going to watch what's going to happen.
Oh boy. He's like, what if I fiddle with my biology some more? What could go wrong?
Yeah. I mean, it is an interesting idea. And kind of the subject of a book that I just read that's coming out in April. Synthetic biology. Genetic engineering. The possibilities are endless.
The future is now. Now, no.
Right?
Now, no.
Dictel's like, is this a joke or just nuts? No,
Yeah.
It's not a joke. He is, he's actually trying to come up with some protocols. So he said, at first, I only thought about it because I imagined the project as purely theoretical. I studied and wrote up a feasible protocol and wanted to post it online for people to see and maybe use. I dug deep and figured out the best place for insertion into the human genome seemed to be this H11 locus, and that one could deliver a CRISPR package in 2 separate viruses. Use CMV-driven SA-Cas9 and U6-driven guide RNA in an AAV serotype dependent on the cell type as they have different proclivities. With a viral titer of 10 to the 11th or greater, you can do injections or intravenous infusions. The more I read, the more I understood how this could actually work, and I became interested in trying to use CRISPR on myself. But that seemed drastic and kind of stupid to try and make permanent genome edits on myself. But maybe I could use all the stuff I learned about studying CRISPR and try to transiently and non-permanently modify my own cells. From what I read, it would only last a few days or weeks at most. I started to work on this part about a year ago, and since then I've explored a number of different techniques to express non-human proteins. GFP, which is a fluorescent protein, RFP, and others in my own cells in my own body. I've been documenting with video recordings that I want to share so that others can learn and experience what it is like to push the cutting edge of science. So yeah, he's been CRISPRing fluorescent proteins into the cells of the skin in his arms.
All right. Mm-hmm.
Yeah, exactly, Whiskey Renegade. He was engineering his skin cell DNA in different spots on his arm. Yup.
Okay.
Yup.
Okay.
Blair, you're just okay.
Okie doke.
So I don't think it's a joke. It's not a joke, Dictel. Josiah is as serious about.
Oh, he's super serious about it.
We interviewed him. He's super serious. Yeah, he is as serious about this as you can imagine. Like, it's not a joke. He is interested in pushing the boundaries of DIY genetic engineering. It's all about biohacking. It's all about the DIY aspect. So garage science, kitchen science kind of stuff. Yeah.
Mm-hmm. Interesting stuff.
He definitely does things that I would never do. I mean, I guess, technically we experiment on ourselves all the time in, various ways, like.
But we're not good about controlling our variables when we do that.
Right. And so he's going about doing this in a controlled scientific manner. So it's, there's the people experiment with their diet all the time. People
Mhm.
experiment with alcohol. People experiment with exercise. People experiment with sleep. People experiment with sex. You know, it's like
Medicine.
I mean,
Drugs.
we are constantly trying things out.
Mhm.
Yeah. But he's just going about doing this in a way that is it's fascinating. Like I said, gonna make my popcorn. Eat my popcorn. And just watch it. Yeah. A little bit of a mad scientist, I would say. He's a punk rock scientist, I think. An anarchist scientist. In that he doesn't like the hierarchical organizational structure of science. He wants to democratize science.
He also likes to skip steps, I think.
Yeah. Yeah, no, exactly. All the experiments that we do, that was the point, Dictel, is that we don't control experiments on ourselves, but
yeah,
here he is. We think, oh, you're doing this crazy thing, but he's actually doing something in a controlled scientific manner, as opposed to the rest of humanity who just runs and goes, blah, you know, no controls, just, I'm going to try this and see what happens. I don't feel too good. Maybe I'll do it again.
What did I eat?
What did I do?
What was it? I should probably eat more ice cream, right?
No, I don't think we did the story about a woman with horse blood replacing her blood. No. Or maybe we did. That would be a long time ago. I don't remember that story. I don't know. Yeah, strength. This is more like body modification as opposed to permanent genetic modification. Yeah, body modding. I'll shove needles in my eyes so I can see more colors. Hey, Blair, have you tried that?
No.
Good night, Dicktail.
And especially considering my aversion to eye stuff, I'm not going to do that. Honestly, one of the only things keeping me from getting LASIK eye surgery is the fact that you have to be awake and they take a scalpel to part of your eyeball.
Mm-hmm.
Nope.
Hey, look, you're going to see it coming.
Yeah, none of that. No, 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 no. No.
Oh my god. All right. I'm going to go. I'm very upset that none of my plans for technology for tomorrow's show are working out the way I wanted them to. But you know what? We move right along.
We'll figure it out. I
It'll be
downloaded
fine.
Periscope on there. I downloaded Google Hangouts. I downloaded Mevo if it shows up before you leave. We'll figure it out.
It's so funny too because I can see exactly where the Mevo is. It's just across a bridge downtown. I can see it.
Yeah, see, I have a package that I was supposed to get today.
Mm-hmm.
It's in San Francisco.
Right?
But it got delayed
due
to weather or something.
Mm-hmm.
And now there's no delivery estimate. But it's in San Francisco.
Yep. That's my package is for 2 days.
Ugh.
Delayed due to weather.
They're like,
tomorrow.
Our delivery trucks can't leave today.
You know what the ridiculous thing is though, is that I have been receiving Amazon packages the entire time.
Yeah, me too.
FedEx trucks have been driving around. I even saw a UPS truck drive right on down. Are they delivering me my packages? No, mine are delayed.
Mm-hmm.
No, I see those trucks. I see them driving. They're driving the streets. Mm-mm.
Guys, stop talking about eyeball injections. I will have to leave. I will literally leave with if you can't stop.
Oh my God. Eyeball injections. No thanks. Know your pandas. You have a panda link.
Yeah, but it's about the better kind of pandas.
Oh, red pandas.
Yeah, and about the convergent evolution between the two. It's pretty interesting.
Awesome.
Yeah.
How are pandas related to ferrets?
Mm-hmm.
That's cool. I'm tired.
Mm-hmm.
I'm tired. And tomorrow I need to do a show. I know. Yes. Brandon, yes. Well, I've ordered the Mevo, so I will be getting the Mevo, and I'm very excited about it. So I'll have it. So for other places and things that we do, not tomorrow, but other places and things, I'll have a Mevo. It's gonna be awesome. I can't wait to play with it. And if you guys haven't seen a Mevo before, you should go check it out. MEVO. They're fantastic for live streaming.
Huzzah.
Hot Rod's waiting for 2 deliveries. Okay, Inside Out. Yeah, that's right. Ed from Connecticut. UPS uses bicycles now. That's right.
Mm-hmm, mm-hmm.
The energy efficiency. Identity 4. Pointed conversations. Oak Tech, looking forward. Oh my God, they just keep going, don't they?
Yeah, enough.
That's
I'm gonna have eyeball dreams now. Kiki, you bring those last 4 calendars? I'm excited.
Should I bring them?
I think so.
Let's see. I think we had one more order today,
okay.
so I need to make sure that I get that order put together. But make sure that I don't. Maybe I can find it here and make sure. I mean, if this person ordered 4 calendars, then we're out.
Right.
No, 1 calendar.
Okay. Yeah.
And I have to bring Justin's calendar.
Right.
Did I reimburse you I reimbursed you your money, right?
No.
I did not?
Mm-mm.
I thought I did. Okay, I'm sorry.
No, it's okay. Let me
do that.
Let me double check. You would have through PayPal like you do your other stuff, right? Okay. Let me just double check. I don't think so. But and then did Patrick get one?
No, I have to mail one to him. I need to
Okay.
get his address.
So just 2.
What? And I
So just bringing 2.
No. The 4
Oh, okay.
without these other ones.
Okay.
But do you have a calendar, or did you order all of them and give them away as
No, I have one. I'm good.
You have it. You're good.
Yes.
Okay, so Justin, Patrick, Fada, and and my dad. I have to give my dad a calendar. So I'll bring 3 calendars.
Great. And I have 1 still to sell. Yeah, I don't think. Yeah, nope.
I mean, technically, I've got calendars because I put these calendars up here. But I kind of like the idea of having a couple of extras to give away maybe later in the year as a little present. So I like the idea of having a couple just kind of held in reserve.
That's a good idea.
Yeah. Oh yeah, California. I'm going to California, where it's like mid-50s and rainy. Do you realize I'm wearing 2 pairs of pants right now?
Oh my goodness.
I've been so cold. I'm like 50 degrees. Oh my God, that's like over 20 degrees warmer than it's been here recently.
Although I don't want to say anything about it because I'm kind of scared. But there's supposed to be thunder and lightning tomorrow. And I'm a little worried for your airplane.
Oh no. Okay, this flight is gonna be fine.
It's gonna be fine.
Fine.
It's gonna be fine.
47. Yeah, go hot rod. Yeah, I think it topped out here at like 41 or 42 degrees today, and it's just icy slush all over the place. It's terrible. It's terrible. It's dangerous and terrible. Don't have free bananas to give out. Yeah, Seattle wet and dim.
Why bananas?
I
don't get it.
Schnago weather. No.
Oh, Amazon gives out free bananas. Why?
I don't understand that. Who do they give bananas to?
Good question.
Mm-hmm. Yeah, Whiskey Renegade. That's what Amazon is trying to do. You're right. Thunder snow. That's a real thing. 10 below, Eric. Oh my God. Oh my God.
Ugh.
I just don't even understand that. I mean, I think our low, we got down to like 8 degrees, and I was like, okay, I'm done here. I can't do this anymore.
Unnecessary.
This is unnecessarily cold. Yeah. Wow. Seattle Amazon banana vendor that just hands out free bananas. Interesting.
Okay.
Mm-hmm.
Anything else for tomorrow? I have your flight info. I'm picking you up. We're gonna come back to my house and wait for Justin.
Mm-hmm.
Then we're gonna head over to the general area.
Yeah, I mean,
and have dinner maybe.
Uh-huh. That sounds like a good idea.
And discuss our stories and plan.
Yeah, I was thinking, what's it called? Home? It's off of Irving.
Oh, that place.
You know what I'm talking about?
Yes.
It's like a block away from the academy.
Mm-hmm. That works. Also, across the street from the academy is Pacific Catch if you want some fish.
That's right. Very good fish. Okay. Yeah, either one would be good.
Home.
Is it called home or I'll find it.
Okay. Social kitchen and brewery.
I mean, it very well could have closed and it could be something new now, but Park Chow. That's what I'm thinking.
Oh, you're thinking Park Chow.
Park Chow.
Yep. Those are all options. Excellent.
Yeah, and it's right
options.
near Pacific Catch.
And
then Nopalito's good also. Mexican kind of.
All good options.
Yeah, I like the idea of dinner. Google Maps, Zoom issues. Fish and chips. I do love fish and chips. 50 below.
How is that even a thing?
Oh my God. Yeah, I don't even know. I know 2 people that are on their way to Antarctica right now.
Wow.
Isn't that crazy? Like most
Well,
years,
that's warm down there though, right?
It's summer, but I mean,
yeah.
It's warm.
It's a balmy 50 below down there. Yeah. My goodness.
Fish and chips. Good night, Identity 4. I'm tired too, actually.
I know. I must depart. I have to work for several hours tomorrow before I pick you up.
Yes, you have to work. I need to pack and get my child to real bed and to real school tomorrow morning, which he's not going to want to wake up for because he stayed up too late. And what else do I have to do? Yeah, and then make sure that I'm showered and appropriately adorned with clothing to go out in public on an airplane and then to do a show. Although I don't even have to do that because I could probably change at your house if I
Oh,
needed
absolutely. Yeah.
Okay. It's all good. Good night, you guys. Everyone in the chat room, thanks so much for joining us tonight. Always fun to see you in there. If anyone's over in the YouTube chat room, oh, it's quiet over there. Quiet over
How do you
on YouTube.
access the YouTube chat room?
You have to go to the live show as it's living. And it has a live chat right next to it. Yeah. It was fun. It was supposed to be a super short show, but we like talking about science. So that's what we did.
These things happen.
And like I said, don't know if we will be able to live stream tomorrow. I will try to social media, let people know. The show starts at 8:15 p.m. tomorrow night. So that's Pacific time. Yeah, so I don't know, probably within the hour before that, I will probably know whether or not we're going to be able to make it work, I imagine.
Yes.
Yeah.
I would say that's
I think they have one
doable.
podcast in the 6-something hour, and then there's a small break, and then we are later. So I think there are just the 2 podcasts, which is good. Yeah, just load the YouTube page. And then, of course, we'll be back next week. I hope everyone has a wonderful night, and we will see you again soon on the interwebs, or maybe in person if you're going to be in San Francisco tomorrow. Can't wait to see you in person, Blair.
Yes, it shall be grand.
Coming to San Francisco.
Yes.
Awesome. All right, I'm going to stop broadcast now. Bye, Kiki.
Bye, Blair.
There we go. Bye, minions.